This is a procedural video for the leaf disc photosynthesis lab. So the first thing you need to do when you get into your groups is figure out which variable you have been assigned. So every lab group is going to be assigned a different variable that they're going to test. You then need to figure out how you are going to test this variable. So for example, you may have been assigned type of light. So you're going to see how different types of light affect the rate of photosynthesis. And you need to establish your control group and your experimental group. So your control group is normal. That's the one you're going to subject to normal conditions. So you'll just set up the lab as instructed in the procedure. And then you'll set up your other syringe. So you're gonna have two syringes, one control, one experimental. For your experimental syringe, you need to figure out how you are going to subject this to a different type of light. You can use natural light from the windows. You can use the flashlight on your cell phones. You can use an actual flashlight, but you need to somehow subject one syringe to the normal light and then another to some other experimental light. And you need to write out a procedure for how you are going to set up your unique experiment to test the variable you've been assigned. The next thing you have to do is you have to actually now set up your syringes to collect data on the rate of photosynthesis. And the way we are measuring the rate of photosynthesis is by collecting data on the amount of oxygen produced. So the way we're going to do that, we're going to take a straw, okay? And you are going to take a spinach leaf. You want a nice dark leaf. Dark leaves are going to have more chlorophyll, and so they're going to be able to do photosynthesis better. And I'm going to make sure I punch holes around the veins. I don't want to punch in the veins because that's going to weigh my discs down. And I'm just going to kind of make sure I get discs like that. And I need 10 per syringe. Again, that is 10 discs per syringe. One, two, three, four, five, 6, 7, 8, 9, 10. And the nice thing about using the straw is that they all wind up kind of just condensed in the bottom there. Next, we are going to insert them into the syringe. So this is the 10 discs for one syringe. Now, you can either kind of squeeze these out like a tube of toothpaste, or the much more enjoyable method of extracting these discs is to just blow in the straw. And they might not come out. But there we go. Loosened them up a little bit. There we go. You got to get them out. Some went flying. Okay, and then you want to tap them to the bottom. So now I've got leaf discs at the bottom of my syringe. Now I need to prepare my solution. Okay, so you're going to take your beaker. I'm going to put in about 300 milliliters of water. It does not have to be exact. Do not spend half an hour of this lab measuring out your materials. So just an approximate 300 milliliters. You're going to use a digital scale to measure about 1.5 grams of baking soda. Again, it does not have to be exact. And then you do 1 to 2 drops of soap. And you can stir with your straw. I'll give you a second to ponder why we've added sodium bicarbonate. Think about why we had to add that to our solution. Okay, so this is the normal. Some of you may have an assigned variable where you need to somehow modify this solution. But first, we need to make sure that we get this normal solution into our syringe. So we're going to push the plunger back in all the way. Don't like press on the discs, leave just a little bit of room. And then you're then going to suck up about enough liquid to fill like 1/3 of the syringe. Again, it does not need to be exact. I'm actually going to do a little bit more. Okay, so that's about half actually. What I'm then going to do, you want to try to set up both syringes at the same time, okay, so that you can start them at the same time. So let's say my variable is that I'm testing the pH of the solution. I'm then going to modify this solution by adding in a couple of drops. Some of you might be changing something else about the experiment and you won't need to do anything to this solution. But if you need to modify that solution in some way, now would be a great time to start this other syringe at the same time. So you want to have both syringes ready to go at the same time, okay? So you might need to modify the solution based on the variable you're testing, and you'll want to try to set up both of your syringes at the same time. So I just have this one set up for now. You guys should be doing both at the same time. I'm now going to take my syringe. You can see there's all those bubbles at the top. I need to get rid of those air bubbles. So I'm going to, just like they do in the movies, flick and squirt. Okay, it's okay if a little bit of liquid comes out. You want to get rid of those bubbles at the top so that there's no oxygen in this syringe. I'm then going to, this is the tough part, why I filmed this video. I'm then going to take my non-dominant hand and I'm going to put my thumb over the top of the syringe and I'm going to hold it. And then with my other hand, I'm going to pull on the syringe like 2 to 3 times. You don't want to pull the stopper out all the way. I'm not taking it out. I'm just pulling it to the edge a couple times while holding my thumb over the top of the syringe. When you look now, the discs will sink to the bottom when at first they were floating, okay? They've now sunk into the bottom and I want to immediately cover this because I don't want to expose it to the light because then the discs are going to start doing photosynthesis. I'm going to then do the same thing to my other syringe, have them ready at the same time, put them under your experimental conditions, turn on the light, put them or set them up however you need to test your variables, start your timer, and you're going to count how many discs are floating per minute. We're going to count a disc as floating if it has risen to at least half the distance of the liquid. Okay, so you're going to leave that liquid in there and you're going to count each minute to see how many discs are floating. You may need to set up your other syringe in a different way. Maybe you're testing the color of the light, the type of light, right? So you need to set up your syringes however you need to. But for the normal one, this is how you would set it up. And don't forget to take pictures or videos or like a time lapse for your presentations.